For most of the 20th century, the story of Richard Feynman's mind was told through physics, quantum electrodynamics, Nobel Prizes, Feynman diagrams, the Challenger investigation. He reinvented how physicists calculate the behavior of light and matter. He cracked safes at Los Alamos for fun while helping build the atomic bomb. He was, by almost every account, the most intuitively brilliant physicist of his generation. His intellect was treated as an individual miracle, a singular brain that appeared in Far Rockaway, Queens, and simply dazzled. But when geneticists began sequencing the DNA of populations like Feynman's, Ashkenazi Jews, they discovered something that no biography ever mentioned. The genius might not have been random, and the evidence was hiding, not in physics, but in medieval cemeteries, population bottlenecks, and mutations that most people associate only with disease. This is the story of what DNA reveals about Richard Feynman's ancestry. And it begins, not with a man, but with a number. 350. Feynman was born on May 11, 1918, in Manhattan. His father, Melville, had been born in Minsk, in the Russian Empire, into a Lithuanian Jewish family. The Feynmans emigrated to America around 1895, when Melville was 5 years old. Richard's mother, Lucille Phillips, came from Polish Jewish immigrants who had settled in New York a generation earlier. Both sides were Ashkenazi, the Jewish population that had taken root in the Rhineland of medieval Germany and later expanded across Central and Eastern Europe. For centuries, these communities were culturally distinct but genetically invisible. Nobody thought their DNA held any particular secret. They were simply European Jews, scattered across the continent, bound by faith and tradition. What reason was there to look deeper? Quite a lot, it turns out. Jews make up about 0.2% of the world's population, 1 in every 500 people. But since the Nobel Prize was first awarded in 1901, at least 220 Jewish laureates have been recognized, accounting for 22% of all individual recipients. In physics alone, the figure is 26%. In economics, 41%. That is more than 100 times the statistical expectation for a population this size. No cultural explanation alone could produce a ratio like that. Feynman himself was one of these laureates, sharing the 1965 Nobel Prize in Physics. His sister Joan became a distinguished astrophysicist. Their father Melville, who never had the resources to pursue his own passion for science, channeled everything into his children. This was common in Ashkenazi families, an intellectual culture stretching back centuries. But culture doesn't explain the scale of the overrepresentation. Something else was at work. And to find it, scientists had to go back not decades, but centuries. 2014, a team led by geneticist Shai Karmi did something no one had done before. Working out of Columbia University with collaborators at Yale, Hebrew University, and Memorial Sloan Kettering, they sequenced the complete genomes of 128 Ashkenazi Jews and compared them against non-Jewish European genomes. The results were startling. The genetic similarity between unrelated Ashkenazi individuals was so acute that Columbia professor Itzik Peyer put it bluntly: everyone is a 30th cousin. The data pointed to a single, devastating event. Sometime between 600 and 800 years ago, roughly 1200 to 1400 CE, the entire Ashkenazi population collapsed to a breeding group of approximately 350 people. 350. Not 350,000. 350 individuals. Every Ashkenazi Jew alive today, all 10 million, including Richard Feynman, traces back to that vanishingly small group. This is what geneticists call a founder event, and it left a mark on every cell. But here is the question nobody could answer from modern DNA alone. What caused this bottleneck? Was it the massacres of the Crusades? The Black Death pogroms of 1348 and 1349? Or something older, something the historical record had missed? Before we see what the next breakthrough revealed, If you're finding this discovery as fascinating as we are, subscribe so you don't miss future stories like this one. In 2013, construction workers in Erfurt, Germany, were converting an old granary into a parking garage. The building sat on unremarkable ground in the center of the city, a patch of earth that had been paved over, built upon, and forgotten. Beneath the structure lay something far more significant, a medieval Jewish cemetery, sealed since 1454, when the city expelled its Jewish population and built over their graves. For more than 500 years, the dead had been undisturbed. Archaeologists conducted a rescue excavation, racing against the construction schedule, and uncovered 47 burials. The skeletons lay in rows, many still articulated, some with identifiable family groupings. And under rabbinical guidance, with specific approval to extract only loose teeth and not bones, a protocol developed by recent rabbinical scholarship suggesting that ancient DNA research is permissible under these conditions. Geneticist Shay Carmi and an international team of more than 30 researchers sequenced genome-wide DNA from 33 of these medieval individuals. The results, published in the journal Cell in 2022, changed everything. These 14th century Jews were genetically similar to modern Ashkenazi populations, but they were more diverse. The bottleneck had already occurred before these people were buried. The disease mutations that define Ashkenazi genetic risk today, mutations in the BRCA1 gene linked to breast and ovarian cancer, variants associated with Tay-Sachs and Gaucher disease. 8 of the Erfurt individuals already carried them. The founding event that shaped 10 million people alive today predated even the 14th century. And there was something else. The medieval Erfurt community contained 2 genetically distinct subgroups. One cluster showed stronger Middle Eastern ancestry, resembling modern Ashkenazi Jews from France and Germany. The other showed more Eastern European ancestry, consistent with historical records that some Erfurt residents had Slavic-given names and origins in the East. The 2 groups had different oxygen isotope signatures in their teeth, meaning they drank different water as children. At least one group were migrants. These 2 populations later merged. And that merger produced the remarkably homogeneous genetic profile of modern Ashkenazi Jews, the profile Richard Feynman inherited. Now here is where the story takes its most controversial turn. If the Ashkenazi population collapsed to 350 people and then expanded rapidly over centuries, the genetic consequences would be enormous. Rare mutations that might vanish in a large population would instead amplify, passed from parent to child, in an ever-growing but genetically isolated community. This is exactly what happened. Today, 1 in every 4 Ashkenazi Jews is a carrier for at least one recessive genetic disease. 1 in 10 carries the Gaucher disease mutation, compared to 1 in 100 in the general population. The list reads like a medical textbook: Tay-Sachs, Canavan disease, familial dysautonomia, Niemann-Pick, Bloom syndrome, cystic fibrosis at elevated rates. For decades, geneticists assumed these were simply the random consequence of genetic drift, bad luck amplified by a small population. But in 2006, Gregory Cochran, Jason Hardy, and Henry Harpending at the University of Utah proposed something far more provocative. They noticed that many of these Ashkenazi genetic diseases cluster around the same biochemical pathways, specifically sphingolipid storage and DNA repair, pathways that modulate early central nervous system development. This clustering, they argued, was too concentrated to be chance. Their hypothesis, published in the Journal of Biosocial Science, was blunt. Medieval European society restricted Ashkenazi Jews to a narrow set of occupations: finance, trade, tax collection, estate management, all of which demanded high verbal and mathematical intelligence. Jews were barred from owning land, forbidden from most craft guilds, and pushed into economic niches where literacy and numeracy meant survival. In this environment, cognitive ability translated directly into economic success, and economic success translated into reproductive success. Wealthier families had more surviving children. Over roughly 800 years, some 40 generations, natural selection pushed intelligence upward. Feynman himself encountered the residue of these restrictions centuries later. Columbia University rejected his application because it had already filled its quota for Jewish students. When he applied to Princeton, the head of the physics department, Henry Smith, wrote to MIT asking bluntly, "Is Feynman Jewish? We have no definite rule against Jews, but have to keep their proportion reasonably small." The barriers that had once driven a genetic bottleneck were still echoing in the 20th century. The cost? The same mutations that may have boosted neural development in carriers, in those who inherited one copy, caused devastating disease in those who inherited two. The evidence was circumstantial but striking. Gaucher disease patients showed a disproportionate clustering in high IQ professions. Carriers of idiopathic torsion dystonia, another Ashkenazi condition, tested with IQs averaging above 120. And the sphingolipid storage disorders, which include Tay-Sachs and Gaucher, affect molecules that play direct roles in brain cell growth and signaling. In a population where intelligence conferred survival advantage, these mutations could have been, paradoxically, beneficial. The pattern was consistent. But was it proof? Here is what the evidence shows. The Ashkenazi genetic disease mutations are statistically concentrated in neural development pathways more than random drift would predict. Here is what it rules out. Simple bad luck. If these mutations had no selective advantage, their concentration in specific biochemical clusters would be extraordinarily improbable. And here is what it implies. That the mind of Richard Feynman, the man who reinvented quantum electrodynamics with hand-drawn diagrams, was not merely a cultural product, but potentially, in part, a biological inheritance shaped by centuries of selective pressure on an isolated population. Not everyone agrees. The drift versus selection debate remains one of the most contentious in human genetics. A 2003 study by Neil Risch and colleagues at the University of California argued that the geographic distribution of Ashkenazi disease mutations supports random genetic drift, not selection, noting that some mutations are concentrated in Central Europe, while others appear only in Lithuania and Russia, a pattern more consistent with multiple independent founder events than a single selective sweep. Others point out that the Cochrane hypothesis relies on indirect evidence. The intelligence link has never been confirmed with controlled genetic studies, and attributing cognitive traits to specific ethnic groups carries a fraught and dangerous history. Meanwhile, the origin of the founding population itself remains contested. Y chromosome studies consistently show that 50 to 80% of Ashkenazi male lineages trace to the Near East, the ancient Levant, consistent with a migration of Jewish men from the Middle East into Europe. But the maternal story is murkier. In 2006, Doron Behar at the Technion identified 4 founding mothers whose mitochondrial DNA appeared in roughly 40% of all Ashkenazi Jews, lineages he believed were of Near Eastern origin. But in 2013, Martin Richards at the University of Huddersfield reanalyzed the same data and reached a different conclusion, that 65 to 81% of Ashkenazi maternal lineages have deep European roots, suggesting that Jewish men who migrated from the Middle East married local European women who converted. If Richards is right, then Feynman's maternal line leads back not to the ancient Levant, but to prehistoric Europe. The debate is unresolved. Both sides have published in peer-reviewed journals. Neither has conceded. What is certain is this. Richard Feynman's DNA was not simply the inheritance of 2 parents in Queens. It carried the compressed history of a civilization, a population that migrated from the Near East into medieval Europe, settling first along the Rhine in cities like Mainz, Worms, and Speyer around the 9th and 10th centuries. a community that survived the massacres of the First Crusade in 1096, the blood libels, the expulsions from England in 1290 and France in 1306, and the Black Death pogroms of 1348 that killed thousands across Central Europe, while maintaining genetic isolation through centuries of endogamy. A bottleneck so severe that 350 individuals seeded a population of 10 million. Mutations that may have been deadly in double dose, but advantageous in single copy, amplified across 40 generations of occupational selection. And through all of it, a culture that prized learning above almost everything else, where the Talmudic tradition made literacy not optional, but sacred. Feynman himself would have found the irony delicious. The man who insisted that nature doesn't care what you expect, whose entire career was built on following evidence wherever it led, carried in his own cells a genetic story that science is still trying to fully read. Today, somewhere in a lab, a researcher is comparing ancient DNA from a medieval European cemetery against the genome of a modern physicist's descendant. The teeth they extracted from 14th century Erfurt still yield data. The sequences still sharpen. If this hidden chapter of history intrigued you, subscribe to explore more mysteries the evidence is only now revealing. Here is the question that remains, not just for Feynman, but for the 10 million people alive today who share his ancestry. If 350 people can seed a population that produces 22% of the world's Nobel laureates, what does that tell us about the relationship between survival, suffering, and the human mind?